with that feeling that they actually understand some of it. As I pointed out to begin, this was not something that I expected everybody to be able to figure out right away. These are not easy to read. Okay, it takes a lot of time to start understanding what the heck is actually going on in here. But I want to start just with this figure again as a reminder of what we're focused on today. So again, the goal of this particular paper is to look at the effects of what? What are they looking at?
How adenovirus shuts down host cell translation.
Bingo. And not only, and we could be a bit more specific than even that, right? We could say shuts down cap-dependent translation. So if we take a look at what we've got up here, recall what makes up EIF4F. What are the components that make up EIF4F? So EIF4E, this is kind of the major one that they talk about in the paper. What else? 4G. What's the role of 4G? Scaffold protein, good. And what else?
EIF4A.
A is what? Helicase, good. And then there are other ones too. So 4G also helps to complex the small ribosomal subunit, and it also helps to complex the poly A binding protein. Okay. I think we'll just start with that. Okay, and we'll see where we go from here. All right. So, skip ahead a few slides. The way that we're going to do this, at least today, is to try and go through each of the figures that I think are most important, and then talk about for each of them what's the question that they're asking, what's the finding that they're making, and then how do they do that. Okay, so we're trying to connect together the question with the methodology, with the results, and the interpretation of this. Let's start very broadly, though. What is the mechanism by which cap-dependent translation is blocked? How does adenovirus do it?
It dumps off or removes the phosphorylation.
Okay, so it prevents phosphorylation of EIF4E, and that phosphorylation is required for the translation to begin. How does it do that? Annie? Okay, so it's got 100K protein, competitively binds with what? MNK1. So it competitively binds to EIF4G, competes with MNK1 for binding to EIF4G. Okay? Starting out the conversation that way today should help to maybe put some of the figures into context. So kind of keep that in mind as we talk about it. Here are some basic facts on adenovirus. It's actually been in the news recently because I'll give the rationale or the reason in a minute, but naked icosahedral double-stranded DNA virus. Genome's about 100,000. I think it's in the 50 to 100,000 range. It's a little bit on the larger side. Synthesizes its genome, transcribes in the nucleus. The name comes from the fact that it was first isolated from adenoid tissue. It causes gastrointestinal distress, and actually there's a number of different potential symptoms. So just recently there was a report that came out that said out of 178 patients that were admitted to a hospital suspected to having influenza, about 10% of them actually had adenovirus infections. So there is this thought that at least adenovirus does contribute to a substantial number of infections, at least in the fall, winter, and spring. We don't have oftentimes don't think about it though. It's not one that causes death in the same way as influenza. Most of the time, the symptoms might mimic something like norovirus or cold, depending on where it's localized. And then the late genes are translated by this process known as ribosome shunting. And this is not one that we've discussed, but one that I'll just kind of bring up a picture to show you what this means later on. All right, so there's our, oh, I guess I'm talking about it now. Ribosome shunting uses something like an iris, but it's not an iris. Remember that for an internal ribosome entry site, that's where the ribosome will bind first for when it's binding to some of these mRNAs for a cap-independent translation. What happens in this mechanism is that you'll get this initial scanning of the 5' end by the ribosome. Oops, now I've done it. And after that scanning at the 5' end, you've got this hairpin structure, something that looks like an iris, that the ribosome just kind of bypasses or shunts past. It kind of skips all these base pairs that make up that hairpin loop structure. This is how adenovirus translates what we call its late genes, those that are produced later on in infection. A lot of those late genes are structural proteins, ones that make up the capsid. But this process here allows for adenovirus to bypass the need for cap-dependent translation in the same way that the host is using it. Do I expect you to know this in detail? No, absolutely not. Just know it's another mechanism that exists for competing with the host for translation. Bless you. Okay. Let's start with figure 1. And the reason why I want to start with this one is just to bring up a couple of features that we've got questions on in the paper. So the first one is, let's review, what is the function of MNK1 or MINC1?
To phosphorylate EIF4.
Okay, so it phosphorylates EIF4E. So in other words, if we have MINC1, EIF4E bound to the cap, it becomes phosphorylated. Where is it phosphorylated at? Sorry, not location. Where on the protein is EIF4E phosphorylated? So they define 2 spots. Annie? Yep. And the other one's right next to it. Threonine 210. Okay, so what we would call MINC1 we would describe as a serine-threonine kinase. And a kinase, again, is just an enzyme that performs a phosphorylation. In this case, MINC1's probably taking a phosphate from ATP, slapping it onto EIF4E to initiate the translation process. So that's what they're showing here. So this is not a Western blot, but it does use gel electrophoresis to separate proteins. And this is an in vitro experiment. In other words, it's not being done in cells. They're doing this experiment just in a test tube where they've added purified MINC1, EIF4E, and there's 2 flavors of MINC1. There's the original or this T2A2. Here they're measuring total levels of EIF4E by Western blot. Here they're measuring total levels of MINC1 by Western blot. This top one here is not a Western blot. There's no antibody being used. Instead, they're detecting P32 EIF4E. What does that mean? What does the P32 refer to?
Radioactive labeled phosphate.
Exactly. Radioactive labeled phosphorus, so you can detect the phosphorylation event. Exactly. So this one is a gel electrophoresis to separate EIF4E and the other proteins that would be in your mix, but instead of performing a Western blot, you're doing what we would call an autoradiograph. You expose, you have a detector that can detect the radioactive material, produces some sort of band where if that radioactive phosphorus is present. So what do we find? Go ahead, Grace.
Oh, I was.
And please ask questions as we're going through this.
They used immunoblot in the article a lot. Is
Ah.
that just Western blot?
It is one and the same. It's a
Okay.
different term. I kind of view it the same way as like people call soda or pop or cola based on where you're at. Immunoblot is like the formal method. So Mink 1 versus T2A2, what's the difference that we observe?
T2 is not a phosphorylate.
Exactly. So it is the same protein, though it's got mutations in it that prevent it from performing the phosphorylation. You'll notice the T and the A here, if I'm not mistaken, refer to amino acids. So the T would be threonine, the A would be alanine. Basically what they've done is a point mutation where they've taken MINC1 and converted it into an inactive kinase. Okay? So it would still be able to interact and still be able to bind to EIF4E, just can't phosphorylate it anymore. In the grand scheme of things, this figure really isn't all that important. But what this is saying to us is if you look at it, the uninfected versus the adenovirus infected, when they pull the proteins out of these cells, there's no inhibition on translation. Okay, so what that at least is saying is that everything seems to be working properly. Yeah, we'll just leave it at that. Let's go to this one though, figure 2. Somebody set up the context for this one. What is the experiment that they're running? Or what is the question? I guess I've written down the question. Annie.
They were looking at the levels of each of the complex proteins in the cytoplasm with and without being infected.
Good.
If, like, the adenovirus is degrading or displacing other.
Good. And so this is a total lysate. So they're looking at, it doesn't matter where the location is, they're just looking at in cells infected with adenovirus or without an adenovirus, are there differences in the levels of these proteins? And all of these, I'm going to do this again, damn it. All of these are part of what? What complex?
Yeah, 4F.
Yeah, exactly. So they're looking at, does adenovirus disrupt that EIF4F complex? So we've got two conditions, uninfected and an adenovirus late infection, which I think they're describing as like 36 hours or so. They infect the cells with adenovirus or not, harvest the cells, lyse them to get the protein, and then they denature the protein so that it becomes linearized and they can run it out. Each of these represents an individual Western blot, so they've probably run 5 gels. And then they probe with antibody that's specific to detect EIF4G, or MINC1, or EIF4A, or E, or poly A binding protein. Just by eye, what's the effect? We could probably conclude that there's no effect. There might be some where you're saying, there might be some effect. Okay, like EIF4G, for example, it looks like maybe in the uninfected the band is a little bit thicker than it is in those that are infected with adenovirus. We can actually quantify the intensity and the area that these bands take up. So we described Western blot as being a semi-quantitative method. We can compare 2 samples and say, hey, there's 3 times more or 3-fold greater amounts in one sample than the other. But we can't tell the exact concentration. And the method to do this is really easy. You just take a computer program that'll tell you what's the intensity in each of those areas. Grace.
So I just had a question.
Mm-hmm.
The way I interpreted late AD infection, I interpreted it as after translation. Is that right? Or?
You are right about that. So what really is happening is this is after the point at which adenovirus is blocking cap-dependent translation.
So like after the MNK1
Yes,
binds.
after the MNK1 binding, or in this case, the displacement of it.
Okay.
Yeah. What would we want to include here? I think that there's something missing from this figure that might help us to determine, is this effect on EIF4G, is this real or is this an artifact of the experiment? Okay, like what, Annie?
Well, that's what I noticed with this. There's not another Western with like a housekeeping protein or something
Yeah.
that you could refer to. Because if you didn't have that, you could be like, well, it seems like a couple of them are actually darker and non-infected. So if you had a loading control, then if you saw that loading control was darker than the other one, then you know that you have that job.
Exactly. So usually going into a Western blot, you'll try to normalize or make it equal so that you've got equal number of cells going into each of those lanes, or you've got equal amount of protein. But sometimes that's not always true. And so what Annie's pointing out is oftentimes there will be a loading control or an additional standard that the levels of that protein in the two conditions really shouldn't change. Common ones would be proteins like actin or alpha-tubulin or GAPDH, which is a dehydrogenase glyceraldehyde 3-phosphate dehydrogenase, something that's involved in glycolysis. And oftentimes then when we're measuring the intensity of bands to determine if they are greater in one condition to another, we set up a ratio. So this ratio would be, what is the intensity of the EIF4G band compared to the loading control? And then we can compare those ratios between the two samples to determine, are they similar or different from one another. Questions on that, on the idea of a control here, or this experiment? All right. Okay, let's move into figure 3. Think about this real quick. Here's what I want to do to start with this one. I'm going to stop talking for a few minutes. What I want you to do with somebody else is identify in A, B, and C what technique or techniques are being used to detect protein in these, and then what is the question that they're asking in each case. Okay? And everybody should start at a different part on A, B, or C, because I don't know if we'll get through everybody, if we'll have enough time for us to analyze all 3 of these before we come back as a group. So each pair pick a different area to go.
Picture of a owl.
Yeah, that one looks scary.
Maybe it's a frog.
All right.
I don't know. Yeah, they have like the chocolate bars that are just filled with caffeine. It's incredible.
What? Okay.
What?
It's like
Wide
one
awake
bar of
coffee.
chocolate is like 2 cups of coffee.
Spectacular.
I don't like them.
All right. So I skipped C on here, so I forgot that I had done that. But I think I don't know why I did, but we'll talk about that one anyway. Actually, no, I do know why I skipped C. Let's go over. Did anybody do A? That's fine. So A is just we said this one is actually another autoradiograph, but they're using radioactive methionine. So they're determining total protein that's being produced. The main takeaway from this, I mean, you could look at this and say, yep, there's a difference, right? So in uninfected cells, you get this big smear of protein, and then as you progress in adenovirus infection, you get less and less, and you get discrete bands. Those discrete bands there are typical for adenoviral proteins. So what this figure is saying is that this is the figure to say CAP-dependent translation is being inhibited, and you've got adenoviral translation becoming the majority of translation that's going on. That's the takeaway point from that image. Go ahead, Jonah.
It's like each of those bands is most likely a viral protein.
Probably a capsid protein,
Okay.
in all honesty, yeah. All right, let's take a look at B. What techniques are being used in B?
We said a Western blot or an immunoblot.
And refer to it as either. It
Okay.
doesn't matter to me. Definitely Western blot. What are they doing before that?
So the GST, is that their marker, right? And then they're just running that as their loading control?
Yeah, the
kinda?
GST actually serves two purposes here. Definitely being used for the detection of MINC1, and MINC1 there will kind of be the control. But what does it mean when they say that they're doing a GST MINC1 pull down?
Does it polarize it?
Doesn't polarize it. What's a technique that we talked about? Maybe another way to ask this is, what are they trying to determine here? Binding partners. What technique did we talk about on Wednesday that analyzes whether two proteins bind with each other?
Precipitation.
This is very similar to an immunoprecipitation. So what they're doing here is, and there's a couple of ways you can do these GST pull downs. Jonah was supposed to do one last semester, but never got to it.
It's not my fault.
Yeah, I know. So newer ways use like centrifugation as a pull down method, or something similar to immunoprecipitation. Older days would be column chromatography, where you'd bind it to a column and then have it eluted out of solution. But they're looking for what binds to MINC1. So given those two techniques here, they pull out MINC1 out of solution. They see what comes with it by performing a Western blot. What do we observe?
The amount of MINC1 produced isn't affected by.
Okay, or at least the amount of MINC1 that they're pulling out of solution is consistent, right? And that's good. That's a really nice control in some ways. How about EIF4G? There's less as infection progresses. What about EIF4E? There's less as well as infection progresses. So those results, what's the interpretation of that, or what are potential interpretations?
So we said that if the adenovirus is blocking the binding site where phosphorylation would occur with both of the EIFs, and then the MINC1 is still there, so it's blocking MINC1, and that's not affecting, it's not affecting those levels, but because it's blocking the phosphorylation, it's blocking the other levels.
Okay, I think we should couple this with figure C real quick. So let's go to figure C, and we'll come back to B here. Shania, you were working on C, right? You and Andrew? Okay. Or if anybody else worked on this one too, this one's an EIF4G immunoblot. So immunoprecipitation of EIF4G, what interacts with it, and then their Western blotting for the partners. What are the observations here? Does EIF4G or its binding to these other proteins change as the infection progresses? No. No, levels seem to be consistent. So what does this tell us then? Let's put these two pieces together, and maybe what we need to do is draw this out a little bit. Okay? I was going to draw it up there, but I think I'll just do it right here. So we know from figure C, we know that EIF4G, regardless of infection, is interacting with E, A. Can folks see that one?
Yeah.
I'll use a different color. I don't like it.
Green.
Black herbal.
Should put a
Green.
green in there.
No,
I feel like it's black and green.
no. Poly A binding protein. Ooh, that's a really nice blue. Very soft. All right. I think that'll be a permanent staple. So it can interact with all of these. And we know that regardless of adenoviral infection, these are always interacting together. What does B say? So what does figure B say?
That the longer infection decreases, we took that as like the phosphorylation of those.
Like dephosphorylation.
So it's
Yeah,
not
the dephosphorylation.
phosphorylation per se in this one, because this one is just a Western blot. It's not even looking at the phosphorylation
True.
status of them. So what? How could we reinterpret that, or what could be an alternative?
Just whether they're binding together.
Exactly, exactly. What that's saying is that here's our MINC1. That's going away during infection. Its ability to bind to EIF4G and its ability to bind to EIF4E is being lost. But that's not because these other proteins in the complex are decreasing in levels. Okay, so basically at this point we know, all right, there's something wrong with the interaction between MINC1 and EIF4E and MINC1 and EIF4G. What's the effect of this?
Translation inhibitor.
Good. Translation inhibition. And what else? What's figure D say? Decrease in phosphorylation of EIF4E. Okay. One of the things to point out here is that none of these images alone tells a full story. Okay. Oftentimes, and I think we kind of hit on this a little bit on Wednesday, oftentimes what we need are 2 or 3 different experiments to kind of help us narrow the focus a little bit more each time to help make the picture more clear, essentially. All right? And in this case, what it seems to be is that loss of MINC1 is leading to a loss in the phosphorylation of EIF4E. Somehow adenovirus is causing that. Questions on techniques, how to interpret the picture, the images, or results? Okay. One quick follow-up question. I don't have it on here, but take a look at figure C real quick. So, one of the bands that they, or one blot they don't run here is MINC1, MNK1. What I want you to do is think about what would the results for MNK1 look like at, in the uninfected compared to those with adenoviral infection. So what would the results look like there for MINC1 as the infection progresses?
We're supposed to discuss it with our partner.
Take a minute to either draw or write something down, and then we'll discuss.
So I think it would decrease because
Yeah.
of not having all of them connected together.
Mm-hmm.
Yeah.
Jonah?
Would the mink one have the radioactively labeled phosphate on it?
I don't think so, because it's transferring it from ATP. If we were to be able to detect MINC1, though, in this experiment here, that's found on figure C, what would happen, or what would we expect to observe?
Decrease.
Decrease. I'm guessing most people would probably argue decrease here, and that fits with what we see in B. We know B, when we pull out MINC1, it's not interacting with EIF4G or EIF4E. We'd expect the inverse too. If we pull the EIF4G out of solution, the amount of MINC1 interacting with it is decreasing over time. All right. And those reciprocal experiments are really nice to include sometimes as well. We're not going to spend much time with 4 here. I think, I don't remember what I've done with figure 4. Oh, there we go. The biggest thing to point out about figure 4 here, let's just focus on A, given the time that we have, because there's one thing that I still want to move through here. But in A, what they're looking at is the ability of translation from a eukaryotic mRNA or from an adenoviral mRNA that has one of these unique hairpin structures. They've taken beta-galactosidase and put it downstream of one of those mRNA leader sequences. S209 refers to that EIF4E that's not phosphorylatable on that site. The S209 T210 refers to the one that definitely cannot be phosphorylated at all. What happens to cap-dependent translation as phosphorylation is blocked? Decreases, and that's what we're observing here. So there's a decrease in that band. And again, this would be cap-dependent translation. So they've taken a cap and put the gene, the LAC-Z gene downstream of it. And the example on the right, the 3' or the 3 leader they refer to is this one that has this elegant structure that looks like a cactus apparently, and put LAC-Z downstream. Fair enough.
Oh, I see.
And what happens to translation of this?
Doesn't change.
Doesn't change, unaffected. Okay, this is how adenovirus is encouraging translation of its mRNAs and while inhibiting the host mRNA translation. We talked about on Wednesday a hurdle that viruses have to overcome is competition for translation. Okay, this is one mechanism, one of many. How does it happen? How does adenovirus block translation? We know that it's excluding MINC1, but how does it exclude MINC1 from that initiation complex?
The 100K protein.
The 100K protein. Right. So the 100K protein is a late gene of adenovirus, again being expressed later on in expression. This is an experiment here, another type of Western blot, but it's being coupled with a different technique, one that we haven't discussed yet today. What does cap affinity purified mean?
Like the strength of like a binding interaction, I guess.
Okay, let's think of it. Let's go back to our original thought, our original image of the EIF4F complex. What does EIF4E bind?
All those.
It
4E,
yeah,
4G,
it binds to
probably.
G and A, etc. But what on the mRNA does it bind? Binds the cap. Cap affinity chromatography. Oh, jeez. A method in which you've got a column for chromatography, and in it you've got beads that have 5' caps. Take your protein lysate, pour it in there. EIF4E is going to bind to the cap. So will anything it's interacting with. And then you elute it out of the column afterwards. What elutes out with EIF4E? And the plus here represents adenoviral infection. The minus means no adenoviral infection. So what interacts with EIF4E?
The 100K protein.
The 100K protein. And, obviously, EIF4G. What would we expect for MINC1?
there to be a line in the not infected and not in the infected?
Perfect. Exactly. Over here on the right, this is what we would call a whole cell extract. So this is just looking at what's the total amount of protein. When we said like cell lysate, that's a whole cell extract or a whole cell lysate. All right, and the flag here is the 100K flag. Remember, we said flag is a tag that can sometimes be added as a way to detect these proteins with antibodies. All right. Just thinking real quick. Let's take a look at B here. This is kind of probably one of the seminal experiments done in this paper to show what exactly is inhibiting this translation. I'm going to give you guys 2 minutes. Come up with an explanation for what you observe in B, and only focus on, don't focus on the pre-immune lane, just focus on the flag and the 100K lane. And notice that they're doing an immunoprecipitation for EIF4G.
They're saying there's flag not present because there's no 100K present. And then on the second one, that the flag is on the 100K, specifically working with MNK1 to block it.
How about somebody we haven't heard from today? What's going on in Figure B here? Somebody who hasn't talked today. Not you, Jonah. Shut up. We should have keys. I think that should be a rule. Like, everybody has a key, and then once you've talked, you can throw it in the middle so you don't have to talk again. I think that's a good idea.
Sometimes I just really like talking.
Yeah, I know you do. All right.
Let's say when the 100K protein is present, then MINC1 doesn't bind to anything.
That's a great interpretation. What are you using to support that interpretation, Andrew?
Like the column on the left, when just the flag protein is present, MINC1 can bind to the rest of the protein group complex, but when the 100K protein is present, that's the only protein that doesn't bind to the rest of them.
Yeah, that's great. One of the things to point out here is in this particular experiment, this is not in the context of adenoviral infection. They're only putting into these cells either, we'd call it an empty vector, so something that has a flag tag and nothing else. And on the right, they're putting in the 100K protein that has a flag tag associated with it. So this one is kind of our control. The 100K represents our experimental condition. And exactly as Andrew pointed out, there's an inverse correlation. One or the other is there, but both of them can't be there at the same time. Okay. We'll go ahead and we'll skip the figure 8. I didn't want to do all of 6 and 7 because it talks about how does 100K actually bind to EIF4G. And that's not something that I think is incredibly valuable for what we're doing right now. But if you wanted to take a look at 8 down the road, the big thing with this figure is they're looking at this again in the context of adenoviral infection. The Ad5-TS1 is a temperature-sensitive strain, one that does not have the ability to block translation. And what they find out in this paper is actually there's something wrong with the 100K protein at that at increased temperatures. Okay. Let's just finish up with kind of closing up the week. So, major things that we have talked about this week. Central dogma, I think, is kind of a key point here. And it's not every little detail. Remember, we've focused on quite a bit polymerases that are required for individual viruses, and that our deciding whether a virus might have a polymerase or not is dependent on genome of the virus, and it's dependent on location of the virus. Where is it actually going to replicate? Where is it going to transcribe? So that's a major theme. The other thing that we talked about was hurdles. How do viruses overcome some of the limitations from host cells? Today's paper obviously focused on how do they overcome the limited number of ribosomes that are present within a cell. How does the virus ensure that its genes are going to be translated while blocking the host translation from occurring? Okay. And then the last part was technique. So we took a look at one of a number of different techniques that are used for not only for investigating viruses, but Western blots can also be used to detect viruses. Sometimes it's used for detection of HIV, and we'll talk about that a little bit more. There's limitations to it, which we haven't really discussed in terms of the diagnostic testing, but really powerful tool if you're trying to analyze protein levels. Okay. Any questions on what we've covered this week? Last minute thoughts on the paper? My quick check. Are you walking out of here knowing more about this paper than you did coming into today? Okay. Go teach a grandparent. That's the true sign of whether you know something. Okay? Okay, but not Brendan's grandma, because apparently she was a virologist. So,
Oh, really?
have a great weekend, everyone.